Grave, authoritative. Typhoid fever is a brutal, systemic infection. Caused by the Salmonella Typhi bacteria, it spreads through contaminated food and water, invading the bloodstream and causing over 6 million illnesses every single year. The burden of this disease is heavily concentrated in densely populated regions with developing water and sanitation infrastructure, primarily across South Asia and Sub-Saharan Africa. For decades, doctors relied on a standard course of antibiotics to clear the infection. But Salmonella typhi is mutating. We are now seeing the rapid spread of multi-drug resistant and even extensively drug-resistant strains. These new variants easily survive our first-line defenses, and in places like Pakistan, they are now surviving our last resort antibiotics, like fluoroquinolones and third-generation cephalosporins. Our medical safety net is fraying. We can no longer rely on treating the infection after it takes hold. We have to stop the bacteria before it ever enters the bloodstream. You might assume that surviving a bout of typhoid leaves you protected. It doesn't. People who recover from the illness remain highly susceptible to future infections. The bacteria achieves this through a highly specific evolutionary adaptation called the capsular polysaccharide. This complex sugar forms a dense outer shell around the cell. When the human immune system sends antibodies to attack, this capsule acts like a biological stealth suit, repelling the response and preventing the body from destroying the pathogen. Protected by this armor, the bacteria can quietly retreat and hide inside the host cells. Because of this, up to 5% of infected people become chronic carriers, silently shedding the pathogen into the environment for years without showing a single symptom. Salmonella Typhi's entire survival strategy revolves around evading immune memory. This means any traditional approach to designing a vaccine starts at a massive biological disadvantage. The earliest attempts to build a typhoid vaccine relied on standard methods. The Ty21a vaccine, introduced in the 1980s, used a live, weakened version of the bacteria to trigger an immune response, but it had a critical blind spot. The Ty21a strain did not express the VI capsule. The immune system learned to attack the underlying bacteria, but it never learned to recognize the pathogen's primary stealth suit. Scientists then developed the VIPS vaccine. This time, they isolated the VI capsular polysaccharide and injected that exact sugar structure directly into the body. The immune system recognized the sugar, but the response was weak. Because the vaccine was made solely of a polysaccharide, it failed to trigger a deep cellular memory, and the resulting protection faded away after just a few years. Worse, this approach essentially failed the most vulnerable demographic. The pure sugar structure was practically invisible to the developing immune systems of infants and toddlers, leaving children under 2 completely unprotected. Science had finally isolated the correct target, but it was speaking a biological language that young bodies couldn't interpret. The breakthrough came with a new piece of biological engineering: Typhoid Conjugate Vaccines, or TCVs. Scientists solved this by attaching the stealthy V capsule to a recognizable carrier protein. The immune system sounds an alarm upon detecting this protein, launching a major attack. This forces the body to build memory cells against the sugar capsule, producing a massive antibody stockpile to recognize the stealth suit. If the actual typhoid bacteria ever enters the bloodstream, the immune system immediately recognizes the VI capsule, strips away the pathogen's stealth advantage, and neutralizes the threat before an infection can take hold. By chemically anchoring the bacteria's greatest defense to a loud biological alarm, TCVs effectively hack the immune system into delivering robust, lasting protection. To prove this immunological theory worked in practice, researchers deployed the vaccine into the exact environments where typhoid exacts its heaviest toll. In large-scale Phase 3 clinical trials across Nepal, Malawi, and Bangladesh, TCVs demonstrated overwhelming success, reducing infections by upwards of 80 to 85%. And because of the carrier protein hack, the vaccine succeeded exactly where previous iterations failed. It provided sustained, robust protection to infants as young as 6 months old. The data out of Bangladesh revealed an additional layer of defense. Vaccinating the children actively reduced the amount of bacteria circulating in the environment, which provided indirect protection to their unvaccinated neighbors. The vaccine functions as a population-level firewall.
It stops immediate outbreaks and breaks the transmission cycle by preventing the bacteria from establishing new chronic carriers. Recognizing the urgency of the antibiotic resistance crisis, the World Health Organization has rapidly pre-qualified multiple TCVs, paving the way for aggressive global rollout campaigns. As pathogens continuously mutate to outpace our antibiotics, these conjugate vaccines represent our best defense. We are defeating a deeply entrenched ancient disease through pure biological engineering.